Method, starring me, Adam, and you.
And me, Ben. Man, I love how we open that. That's great. So my name's Ben Edwards. I'm a university student studying biology.
I'm Adam Simpson, and I'm an undergraduate studying neuroscience. So the method, named after the scientific method. Ben and myself, we're trained to work and live by this method. Honestly, the aim of this podcast, we just want to educate people, create some interest in science, and
make it fun.
Yeah, make it fun, and just have a little discussion on important topics in any scientific field, past, present, or future.
Yeah, so we'll be talking about biology, maybe some physics, some cool space stuff.
Yeah. Anything really.
Who knows?
Sky's the limit. So, what we're talking about today, actually, CRISPR. You may have heard the word CRISPR thrown around a lot in media, but what exactly is it? Yeah, we're still delving into it, but essentially, the Legos of life. We're toying with the Legos of life. You may know. Yeah, your genetics. They're the building blocks of what organisms essentially are made of, and it's what makes you, you. And CRISPR is essentially allowing us to take those apart, put it back together, change things around. Pretty wild stuff.
Well, let's get into the meat and potatoes of this thing. What exactly is CRISPR? So it's an acronym, actually, spelling out CRISPR, standing for Clustered Regularly Interspaced Short Palindromic Repeats. And these genes are of similar composition found within the genomes of bacteria that aren't necessarily native to the bacteria's genome.
Yeah, so essentially CRISPR almost acts like a bacteria's immune system. A virus comes in, starts trying to attack bacteria. The bacteria essentially takes the DNA from attacking viruses and sort of assimilates it into its own immune system. Similar to how humans work, we take certain infections and whatnot, and we sort of tag them, remember them for future reference, and make sure that when we encounter these sorts of viruses or infections, what have you, in the future, we can essentially identify them before they destroy us, before we know what to happen.
Right, and once we identify what they actually are, we can deal with them effectively and thoroughly.
And CRISPR is essentially, it's a simplified form of it. It's almost rudimentary. It's something that we looked at in bacteria and thought, hey, maybe we could use this in a human sort of function.
Right. So why exactly are these CRISPR genes important? So the main component of this technology is Cas9. This is standing for CRISPR-associated protein 9. And this protein is a protein that acts as a knife, cutting DNA at specific spots determined by a piece of RNA that guides the knife to where you want it to cut.
Yeah, and RNA, some of you may not know, it's very similar to DNA structurally, but it's just a single strand of genetic code that is essentially a copy of your DNA, a functional copy, if you will. And instead of risking our DNA, dragging it out of the big old center nucleus of our cells, you want to keep that stuff safe. It's the master copy.
Yeah, you don't want to lose that, because if it's gone, then SOL.
You don't have anything to work with.
Yep.
And so essentially RNA, we're able to, make a copy of our DNA, put it onto this strand. And RNA is mostly used in things such as creating proteins. It's, what activates all the processes in our body. Mm-hmm. And so RNA is a single strand, essentially copy of DNA.
This protein is a main component in the bacteria's immune system, like we've already said, allowing it to quickly use CRISPR genes to code the immune proteins to recognize hostile DNA. It's a self-contained system and therefore can be used in any organism you want to put it into. Like we already mentioned, you could put it in humans, different animals for, maybe changing their genetics, like pigs. We could use their organs as potential transplants for people who need them.
Yeah, and the fact that it is a self-contained system makes it sort of easy to transplant it from bacteria into any sort of other function or use. And that's what makes it so easy. So when people discovered this, they looked at it and say, hey, we can take this straight out and use it in our own regard.
Yeah, that was back in June of 2012, actually. There, among other researchers, but Emmanuel Charpentier and Jennifer Doudna posted published rather, an article in the Journal of Science. They're from the University of California, Berkeley. And they reported that using previously found information on these proteins, they worked out that they could fuse two of the smaller components together, making a single synthetic guide, further simplifying the system and making it so we could effectively throw this system into eukaryotic cells, so that of larger animals like yourself.
Yeah, and it's important to remember that these components, they're all more or less different types of RNA or different proteins. A lot of the biological processes that take place in your body are just machinery, essentially, of all these proteins, RNAs working in tandem to generate whatever it may be, proteins, more proteins, or just, again, copying RNA is even a prime example.
That's a really important point to make, is that these processes aren't magic. Like, they're literally machines. It's the same as your computer. It's the same as a clock.
Yeah.
If there's certain chemical reactions or protein interactions, then certain things will happen. It's not just...
Yeah, a lot of functional moving parts to
for
this
sure.
thing. So just going to talk about some of the current research. It's relatively recent. 7 years can be a lot in science, but with CRISPR, we're taking baby steps.
Yeah, but they're big baby steps.
They are big baby steps.
We just finished the Human Genome Project back in 2001, I believe.
Yeah.
And we're already going into the fundamental building blocks of our genome and changing them.
Yeah. And one of the literal most current recent developments, beginning of this month, November 2019, first trials in USA regarding CRISPR gene editing. It's complicated and it's surprising because we don't get federal funding in the USA from the government regarding gene editing due to, something you don't want to touch even with a 10-foot pole regarding ethics. A lot of people don't understand it. But
no, it's definitely hard to have an opinion about something that you're not well educated in.
Yeah,
it'd be easy if you're a policymaker and had a background in CRISPR or genetics, but the majority of them don't, and that makes it a really tough issue to tackle for them in terms of making regulations and everything of the sort, which is exactly where the co-founder of CRISPR comes in.
Yeah, and she has kind of spoken out, make sure...
Yeah, Miss Doudna.
Yeah, she's sort of forewarning everyone, we should pay attention to the regulations, we shouldn't dive headfirst into this stuff, know what we're doing, what we should be doing with it before doing it.
Yeah, be smart.
And these first trials, only using a couple patients at a time. I believe only one patient with beta thalassemia, one patient with sickle cell disease.
These are both diseases to do with your blood and malformed hemoglobin, which allows you to bring oxygen from your lungs to all the cells in your body.
Both genetically based diseases, which of course is where CRISPR can come in. And honestly, the trials have been tremendously successful. In under a year,
it's crazy.
Both patients have seen extreme dramatic effects of their conditions. They haven't had to do as many blood transfusions for the beta thalassemia patient. And the sickle cell disease patient has experienced, a lot less, crises involving complications of the disease.
And for those who are interested in how they went about this, they went in and extracted parts of, or some of their bone marrow. And then they went on to use chemotherapy to kill off the remaining bone marrow that they had, which is pretty regular for people who have bone cancer. Then they went on to genetically modify the cells that they took from the bone marrow and then reintroduce it into the patient. And now they're forming proper hemoglobin, so they're able to transfer oxygen from their lungs into their cells effectively and not die at an early age.
Yeah, and essentially that chemotherapy is sort of the blunt part of the treatment.
Right, it's tough for sure.
You're probably wondering, any side effects? What's the catch? The chemotherapy was honestly the brunt of it, and a lot of people go through that as it is. So it's kind of a small price to pay in terms of, the long run. If you have to stop getting blood transfusions, like 16 times a year or something, it takes all that out of the equation.
So the people who are doing this research is a U.S. giant called Vertex Pharmaceuticals, and they're working in tandem with a Swiss company called CRISPR Therapeutics. After releasing these results from the study, they have decided to make another milestone. They wish to cure muscular dystrophy, actually, which is a big thing. Yeah. Because lots of people deal with that, and it's tough. Yeah. There's no cure for it at the moment.
Yeah, and very similar vein of similar to cancer. Cancer and muscular dystrophy aren't just these, singular things that people have. They're very complicated diseases, and there's many variations on it. And, if, we can help one person with one type of muscular dystrophy or cancer, usually the rest of them can come down the line a little easier.
Mm-hmm. So currently, some of the most interesting and relevant work being done with CRISPR-Cas9 has to be with gene therapy, like we've been talking about.
Yeah.
Like, using this technology, we're testing your ability to treat various blood disorders like sickle cell anemia and beta thalassemia, like we've already spoken about, as well as different types of cancer, and facing challenges associated with different animals as viable sources for transplantable organs.
Yeah, and we're barely scraping the surface of this.
Yeah, we sure are. And on top of that, I know lots of people have opinions about GMOs. Mm-hmm. However, this could be a breakthrough for making crops more viable, making them produce more nutritional food for people. Yeah. Making them grow quicker.
Yeah, and there's, all risks even put into that. Things look up when you have this much potential at our fingertips now.
For sure.
No one would have ever foresaw this sort of thing happening in the past decade. Mm-hmm. Yeah, so, of course, combating cancer and everything it's our primary objective right now. But in the future, there's some, stranger things that we could think of using this for. The possibility of reviving recently extinct species.
Yeah, that's a big one.
That's bizarre.
It's huge.
Yeah.
Like, the blood found in that mammoth, however many years ago that was, it was less than 10 years ago.
Yeah.
But it's crazy to me that we could go in, take that genetic material, and
we could toy with it. And I mean, even using CRISPR alone, we could create new species essentially that are distinct from one another. It's extremely complex, but considering how many people are paying attention to this, all eyes are on CRISPR for the past, half decade now. It was only 7 years ago that they discovered this now, and, people have been running rampant with it. So, essentially any genetic condition is going to get their turn at some point, other than cancer and everything, sickle cell disease, cystic fibrosis, hemophilia, all these things are going to be in line for CRISPR.
Right. And I think that's a good segue into talking about the concerns about CRISPR.
For sure.
So, like we've said, there are major ethical concerns to do with CRISPR within the scientific community and the general public, because it's a scary thing to think that somebody could go in and change anything about anyone.
Yeah, for sure. And especially in medicinal research with children, do children even understand what this would be? Mm-hmm. It's a tough thing to even explain to someone who's not in the scientific community, which, it goes beyond the fact that CRISPR's got so many uses. It makes it a policy nightmare for governments to decide whether or not, is this safe? Should we be doing this at all? Mm-hmm. It's going to be hard to get the general public on the same page.
It is, especially with how imprecise it is at the moment.
For sure.
Like, we've made major strides in terms of how accurate it is and how consistent it is in cutting certain genes where you want them to be cut. But we only have one person's genome to work off of at the moment. Yeah. We're not, or currently the people who are working with it aren't taking aren't sequencing the genomes of every single person that they do it with. Mm-hmm. And that causes some imperfections in the system.
Yeah, or even touching back to Cas9. Cas9, there's not one specific type of Cas9. There's actually variations on Cas9 in which some may be a different size from one another, but then one might be more precise than the other. And it's just a matter of finding the proper machinery that can give us the best results, the most consistent results, that essentially alleviates most of the risks of imprecise cutting.
Right, because if there is a mutation, for example, at the site that you want to cut, and the CRISPR the marker that you've generated, the RNA marker for the gene that you want to cut, if there's a mutation there, then it won't cut at that spot.
No, and worse, it could cut somewhere else.
It could cut somewhere else, which is
not
a good time.
Exactly. We're not even quite sure what would happen. It could, open up a pit.
It could. But also, on that note of opening a pit, if it does turn out that something else opens up or something else happens, rather, then that's a good starting spot to jump off of for figuring out what exactly these different places on your genome do.
Yeah, for sure, because we're not even sure, considering how everyone's genome is very distinctive.
Yep.
And, most of the medical history and things that we know in the science community are from people who come out of the woods and say, hey, I got this thing wrong with me. And then you look at them and you see what's different in them compared to a neurotypical person or something like that.
Yep.
That's what that thing does.
Yeah.
And, there'd be probably some consequences that happen like in CRISPR, like most things.
For sure.
But, like we've said regarding current research, the U.S. actually prohibits funding to gene editing research for a lot of these reasons.
Yeah.
There's bioethics are a nightmare sometimes. Not in every part of the world, though. China is going crazy with CRISPR.
Yeah, they're really going for it.
Yeah, China really dives headfirst into a lot of some of the researchers in China actually have done research and not announced it till afterwards to the rest of the world. Much to everyone's, I don't know, horror / amazement.
Yeah, it's tricky because you want to say, hey guys, you shouldn't be doing this at the rate that you're doing it.
Yeah.
But also, what are the results?
Yeah, what did you do?
Yeah.
Did it work?
Did it work?
Yeah. And, just amongst the general public, if you tell someone that you can go in and edit genes, people are going to ask, where do you draw the line?
Yeah, it's scary.
Are we playing God sort of thing?
Are we playing God?
Yeah.
That's the big
one.
That's a big question.
Yeah, and I don't know.
Maybe a little bit.
Yeah, I don't even think we're ankle high in CRISPR.
Oh, not at all.
Yeah.
The possibilities are seemingly endless.
Yeah, so I think it's important to take away that CRISPR is not going to be some sort of everyone's designer baby nightmare hell, and it's not a cure-all for anything either, but it's very promising.
Very.
Yeah, and it's definitely something. Keep your eyes peeled and your ears peeled for any future news regarding CRISPR, because if you haven't already read some about it, you're going to be probably hearing a lot about it.
Yeah. So if you guys want to check out any other material that we have regarding CRISPR, head on over to our Reddit, or our subreddit rather, at r-TheMethod.
You can find us on Instagram at _method_podcast. We also have a Twitter account at method_podcast. So if you want to send us any sort of questions regarding episodes we have done, maybe even encourage some, episode suggestions, we're open to any ideas. And Ben and I are looking forward to spreading more of the good word around.
Good word. Cheers, guys.